trainees that were going to be on the call today, I wanted to talk about a project that kind of developed out of that transition from trainee into being a PI. So for those that don't know me, my lab studies various aspects of vascular cell biology, particularly things that contribute to endothelial cell activation in atherosclerosis, as well as smooth muscle, mechanisms of IR injury, signaling by reactive sulfur species. And I'm going to talk to you today about what I've often referred to as my ugly dog project, partially because it is one of my favorite projects. I love this stuff, but it has been really difficult convincing anyone else that it was worth studying. So I worked on this for 10 years, and I submitted 6 different R01 applications before it got funded. And you can see the results of those: 42%. 14% with a 12% pay line. That was brutal. Not as brutal as getting not discussed twice after that. Then 29%, and finally a 2% about a year and a half to 2 years ago. So this project came about from the main project in my lab, right? So I started off as a postdoctoral fellow working with Martin Schwartz. And we were looking at some of the signaling pathways that regulated endothelial activation with shear stress. And these are things that regulate integrin signaling and cytoskeletal remodeling and those types of things. So when I was starting off, in my own lab, I didn't know whether me and Martin could both exist in that space. Luckily, we've been able to figure it out. But not knowing that, I was looking for something new to work on. So I was curious if there are any other stimuli besides, shear stress that affected endothelial integrin signaling, cytoskeletal remodeling that may be playing a role in atherosclerosis that people hadn't described there yet. And that's when I started looking at the neuronal guidance molecules. So these are things that have been known to affect cell adhesion, cytoskeletal remodeling pathways during migration and angiogenesis and vascular patterning. But there really wasn't hardly anything known on atherosclerosis whenever we started doing this. So this family is really 4 different groups: the slit-robo pathway, the netrins, the semaphorins, and then the F-ephrin receptors. There had been several studies suggesting a role in inflammation, but very little in athero. That changed as we were developing the project, as things were coming out. Several groups started getting on this topic. Catherine Moore's group published several studies on the guidance molecules in athero. And the basic idea was that in endothelial cells, this ephrin B2 affected leukocyte recruitment, whereas this other protein, SLIT2, limited that. And netrin and semaphorin 3E within the plaque affected migration of the macrophages out of the plaque, right? So both of these are adhesion migration phenotypes, right? So I decided to focus my work in on the F-ephrin family. So these are receptors that are on the surface of the cell. Their ligand is also a cell surface ligand. They're named for the erythropoietin producing hepatocellular carcinoma, EPH, and their ligands are F receptor interacting proteins. So that's where ephrin comes in. In vascular biology, this has generally been restricted to studies on ephrin B2, FB4. Ephrin B2 is a classic marker for arteries. FB4 is a classic marker for venous endothelial cells. So this is really surprising to me because this is actually the largest family of receptor tyrosine kinases in the mammalian genome. And they have different ligand specificities. Generally, the A receptors bind the A ligands, the Bs bind the Bs, but that's not always the case. So, if you look at these, people hear neuronal guidance molecules, they think neuronal, right? They think that this is going to be involved in the brain and it may not be involved in their tissue of interest. That's not the case. right? Here is just the expression pattern for the A family, and you can see that there are several receptors that are expressed generally across a lot of different tissues. Here's brain tissue here in the yellow bars. You can see several of these aren't really even expressed in the brain, right? At least not to any appreciable level. So to start this problem, my first graduate student, Stephen Funk, who's now an instructor at WashU, asked the very basic question: what of these receptors do endothelial cells express, right? Since we were focused in early on the endothelium. And we saw between a couple different endothelial cell lines that it was Fa2 and Fa4. When we treated endothelial cells with pro-inflammatory cytokines or oxidized LDL, we saw A2 going up, whereas A4 didn't. So we focused in our work on A2 and to see how that may be affecting endothelial function in athero. We started looking through the literature. There was actually quite a bit known on Fa2 in the areas of angiogenesis, tumor genesis, particularly in breast cancer, as well as in inflammation, regulating cell-cell interactions and things like LPS-induced lung injury. But there was nothing known on this in athero. So we asked the very basic question: what cell types within the plaque express Fa2? So we took a early fatty streak from a mouse, more advanced human lesions shown here. And you can see PCAM in blue labeling the endothelium, MAC2 in green for macrophages. And Fa2 appeared to localize pretty strongly to the endothelium, but also in the macrophages within the plaque, whereas the underlying quiescent smooth muscle cells really didn't express much. Similarly, within the plaque, Fa2 was not expressed in the media at all. The endothelium overlying the plaque showed strong positivity, and there were a lot of positive cells within the plaque as well, some of them co-localizing with CD68, suggesting that they may be in the MAX2. So to see what this was doing in endothelial cells, the first thing we did was look for the ligand, right? We know the receptor is there. What about the ligand? So when we treated with TNF, IL-1, OXLDL, all of them induced the classic Fa2 ligand ephrin-A1, albeit with different time courses. And then to see what that may do, we took our endothelial cells and treated them with recombinant FC ephrin-A1. We did a QRT-PCR array way back when, and that pulled out a number of pro-inflammatory genes. One of those we went to validate VCAM, and you can see a nice induction of VCAM1 expression here when we treat with ephrin-A1. And when we knock down Fa2, that induction is depleted. In addition to promoting endothelial pro-inflammatory gene expression, we thought since Fa2 is present on the surface of these cells, maybe it's mediating part of that interaction between the monocytes and the activated endothelial cells. So we did a couple of fairly simplistic assays. We took monocytes in culture, plated them out on recombinant ICAM, VCAM in the presence of ephrin-A1 or Fa2, and we saw that Fa2 could increase the monocyte adhesion in vitro. In addition, we saw Fa2 increase the monocyte spread cell area, right? So when we plated them down when Fa2 is present, they were able to spread a bit better, suggesting that this interaction between the monocyte and the endothelial cell may enhance the sticking of the monocyte to the surface of the plaque. And that was gained further support when we stained for the activated receptor, right? So this is a stain for phosphorylated Fa2 here in blue, and you can see this macrophage bound to the surface of the plaque, and that macrophage is where you see the strongest blue phospho Fa2 stain. So at this point, another grad student, Alex Finney, joined the project. We used an ApoE knockout, Fa2 knockout mouse, just to give us a basic idea of what this was doing in the plaque. And we treated for 8 weeks, 12 weeks, 16 weeks, and at every time point, we saw a nice reduction in plaque area when Fa2 was depleted. We also looked in the aortic root, which I don't show, as well as in the innominate artery and the carotid sinus, and in every vascular site, plaque size was smaller. This also correlated with a reduction in macrophage area within that plaque, consistent with a reduction in early inflammation in the plaque. So, we started looking through the literature on, any links between Fa2 and atherosclerosis susceptibility. And the Fa2 gene is actually in a region of chromosome 1, 1p36, that is linked to premature MI. It's in a region of the mouse genome on chromosome 4 linked to enhanced susceptibility to atherosclerosis. And 2 years ago, a SNP was discovered that links Fa2 to coronary artery disease in humans, but we didn't know that whenever we started the project. So when Alex really started kicking off her project, she was very curious about the role for monocyte macrophages, right? So what Fa2 could be doing in the MACs. And this became complicated, right? Because when we started to look for Fa2 signaling in the macrophages, we started using bone marrow derived MACs. And we quickly figured out that bone marrow derived MACs did not express our receptor. In fact, we were able to see good Fa2 expression in human peripheral blood monocytes, in mouse peritoneal macrophages, and in the raw macrophage cell line. But in more pre-monocytic cells, like the THP1s, Fa2 was gone. And in the bone marrow derived MACs, Fa2 was gone. And the THP1s and the bone marrow derived MACs actually show a pretty similar expression pattern, which kind of suggests to us that perhaps there is a change in FFRN expression as the macrophages are migrating out of the bone marrow and maturing. We haven't actually been able to go after that, but that's just an observation we made. So when we started looking at the mouse peritoneal MACs, Alex failed to see any effect on macrophage gene expression, macrophage lipid uptake. So we did a bone marrow transplant just to see if we could see any effects with macrophage versus tissue resident cells, treated Western diet for 12 weeks. And we really saw the strongest protection when the recipient was the Fa2 knockout, right? So even wild type macrophages in a Fa2 knockout, we saw a pretty good reduction in plaque. And if you look here, I think this is the innominate, you see a nice reduction in macrophage area there when the recipient mouse is deficient for Fa2. So we think it's at least in the early plaque, the resident cells confers the greatest protection. So just to summarize part 1 so that everyone's on the same page, we showed that endothelial expression of the Fa2 receptor and its FRNA1 ligand occurs under pro-inflammatory conditions and actually contributes to endothelial pro-inflammatory gene expression and macrophage recruitment. We showed when we deleted Fa2 from the plaque, we saw a decrease in plaque associated with a decrease in inflammation, and our bone marrow chimeras suggest it's the resident cells that mediate that. So the story became more interesting whenever we started looking at some of the more well-developed plaques. So we had these plaques scored by a pathologist using the STARI scoring system. And at 8 weeks, even though the Fa2 knockout plaques are smaller, the plaque grades are pretty similar, right? But when we let that go to 16 weeks, we actually see a decrease in the amount of plaques that transition to the higher grade plaques that are more associated with smooth muscle incorporation and matrix deposition. And then when we looked at some of those plaques for smooth muscle content, standing for SMA or smooth muscle myosin heavy chain, those were significantly down in the Fa2 knockout plaques. So what is known about Fa2 in smooth muscle biology or smooth muscle phenotype within the plaque? So normally smooth muscle cells are in a quiescent contractile state. However, in the of athero, they undergo a phenotypic transition or they lose some of those contractile markers, they migrate into the plaque, proliferate, start to deposit extracellular matrix. So looking at some of the more advanced plaques, we co-stained with Fa2 and markers of smooth muscle cell, smooth muscle actin, and we could see co-localization between SMA and Fa2 within this plaque, both in the mouse and in human atherosclerotic plaques. But what I think is particularly interesting with this stain is if you look at where smooth muscle cells are actually undergoing the phenotypic modulation, it's going to be down here in the, can you guys see my cursor? Yeah, it's going to be down here in the medial layer immediately underlying the plaque where you start to see the loss of SMA stain, right? So when you're starting to see this loss of SMA in these medial smooth muscle cells, that's actually where you start to see Fa2 coming up in the media, right? So it looks like Fa2 is expressed as they transition from the contractile to the synthetic state. And when we mimic this in culture, by taking smooth muscle cells and putting them under serum-free conditions and then hitting them with serum, we see that as the smooth muscle cells lose SMA expression, Fa2 expression comes up, right? So Fa2 is coming up only in the synthetic smooth muscle cells. Then if you look at some of the other family members, Fa2 is the only member of the FA family that goes up when they go through that transition. FB4 actually goes up as well. That's a protein that Alan Dardick has studied quite a bit in smooth muscle phenotype in things like vein graft restenosis. So it's not surprising to see that this receptor would be going up in the smooth muscle cells undergoing that synthetic change. If you look for the FA ligands, you really don't see much there, right? So, we're seeing the receptor coming up, but not a lot of ligand expression, especially not a lot of expression of FRNA1, which is the main ligand for Fa2. So we were curious how Fa2 was regulating smooth muscle cell function. So Alex did a couple of experiments where she just overexpressed Fa2 in the smooth muscle cells and started looking for those phenotypic markers. The first round, she did a MOI of 50 and saw a nice drop in a lot of the markers. I told her that was way too high and we needed to recapitulate that at a much lower level. So we've gone back and done a couple of ends. There's a couple more that are down that are hopefully will round out this data, but we see the same trend, but we haven't hit statistical significance with a much more reasonable MOI of 3. We did some RNA-seq with these smooth muscle cells, either taking them under the quiescent conditions and overexpressing Fa2 alone. So what was Fa2 sufficient to do? And then taking them under the synthetic condition, knocking down Fa2 to see what it was required to do. And under both cases, we see expression of genes that are involved in cell movement, as well as cell survival and cell proliferation. And if you look at the genes that are up in the overexpression or down in the siRNA or vice versa, you see a lot of proteins that are associated with proliferation, such as Ki67 here, right? So that's their classic proliferation marker. So when we looked in the plaque for Ki67 staining, we actually saw a strong reduction in the Fa2 knockout. And this was both in the cells within the plaque, as well as cells within the adventitia. So we think there's a nice reduction in proliferation across multiple different cell types, likely. If we looked at our smooth muscle cells in culture, so here we have smooth muscle cells under the serum-free conditions. You don't see hardly any proliferation here. You hit them with 1% serum, proliferation goes up. But when we knock down Fa2, we see a nice reduction in proliferation, both using Ki67 as well as BRDU stain to label the proliferating cells. And then this is your classic, wound healing assay where we, serum starve, hit them, 18 hours, and you can see with, mock, we got a nice filling of the wound. When we deplete Fa2, that's down. So, being from a signal transduction background, I think the signaling aspects of Fa2 are one of the more interesting parts of its biology. Normally, you think of a receptor signaling in the context of ligand, right? Fa2 can do both ligand dependent and ligand independent signaling. So, in the presence of a ligand, typically on an adjacent cell, you'll get phosphorylation on a couple of different tyrosine residues. And this actually inhibits, at least this is work done in the cancer models, right? It actually will inhibit mitogenic signaling pathways like AKT and ERK, which turns down proliferation and migration. Migration, it acts as a chemorepellent, right? So as two cells, hit that don't want to be together, it'll cause the cell to turn around and go the other way. However, in the absence of ligand, you can get phosphorylation through a couple of different pathways on a serine 897 site, right? And when this occurs, it's known in the cancer field to have an opposite effect where it increases proliferation and migration through unknown mechanisms, right? So, we were curious, how Fa2 was signaling in the context of smooth muscle cells in atherosclerosis. So when we hit with 1% serum, which is the stimulus that we use to induce proliferation, we see an increase in the serine 897 site, right? So increase in this ligand independent signaling effect, and we don't see any induction in the classic ligand induced signaling 588. 772 is up a little bit, but we don't think that's ligand dependent, and I will tell you why in just a second. This work has been picked up by Matt Scott, a postdoc in the lab, who's really looking to answer the question of how this signaling is occurring. Consistent with our idea that this is all ligand independent, when we hit smooth muscle cells with serum, we actually see a decrease in that ephrin A1 expression that occurs fairly rapidly and is sustained for several hours. So it looks like that is, at least that transition is going to be promoting a ligand independent signaling pathway. And then when we look at the smooth muscle cell response to serum, all right, this is you're talking serum, it's a potent mitogen, right? So when we hit our wild type cells, and this is Fa2 wild type, and this is not knockout for some reason, I keep forgetting to put those labels in there. So ERK comes up and stays up in the wild type. But when Fa2 is gone, it comes up the same amount early, but then drops down late. So you don't get the sustained activation of ERK. And for those that follow the mitogenic signaling pathways, that sustained activation of ERK is actually critical to stabilize CFOS expression and to drive a lot of those cell cycle genes. So a transient induction won't do it. And then we look to see if this was sustained after 24 hours in 1% serum, and we do see the change in ERK and the change in AKT sustained under those conditions. We looked at phosphorylation of ERK and phosphorylation of AKT within the plaque, both total plaque area as well as localized to the smooth muscle cells. And under both conditions, we see that those are reduced in the knockout, consistent with what we had shown up here. And then if you look for that S897 phosphorylation site, right, that ligand independent site, we actually see that localizing quite well to the smooth muscle cells that are here migrating in, as well as the smooth muscle cells that are undergoing that phenotypic modulation down here in the media. So what about the other side, right? I mentioned that smooth muscle cells don't express a lot of ligand, but smooth muscle cells are not the only cell type present in the atherosclerotic plaque. Macrophages express a ton of ligand. So, it's possible that interactions between the macrophage and the Fa2 receptor on the smooth muscle cells will have the opposite response. It'll induce ligand dependent signaling. So when we hit smooth muscle cells with exogenous ligand, we see phosphorylation of 588, 772, like you would expect. But when you look at activation of ERK and AKT, you see the opposite response. So ephrin A1 treatment reduces ERK phosphorylation within the smooth muscle, reduces AKT phosphorylation, and if we block Fa2 activity with this inhibitor here, that reduction is lost, right? And if you look at proliferation of the smooth muscle by Ki67 staining, FcRNA1 treatment reduces smooth muscle proliferation. So we think that there are two different effects going on here. One, when these smooth muscle cells are, just expressing the receptor by itself, you can get activation of this ligand-independent pathway that drives proliferation. But two, there's the potential for ligand-expressing cells to ligate this receptor and cause the opposite response to turn the proliferative response down. So to summarize part two, enhanced Fa2 expression during smooth muscle phenotypic modulation promotes smooth muscle proliferation. We think that it's ligand-independent signaling through S897 that mediates this effect, whereas Fa2 ligation with FRNA1 may cause the opposite effect to reduce proliferation and mitogenic signaling in these cells. So the other interesting thing that we saw in the Fa2 knockout plaque was a reduction in fibrosis. Right. So we see, if you look for trichrome stain here, as well as polarized picoserous red staining, you can see the plaques from the Fa2 knockout actually have much less collagen deposition in them. And if you look for fibronectin deposition, which is one of the proteins my lab studied for quite some time, that's considerably down as well. And we found that interesting for a couple of different reasons. One, fibronectin promotes smooth muscle phenotypic modulation. That's known to occur. Two, when you delete fibronectin from mice, you reduce smooth muscle incorporation within the plaque. That was work by Reinhard Fassler's lab at Max Planck. And three, fibronectin plays a critical role in the deposition of other extracellular matrix proteins. So if fibronectin is not there, the ability of those cells to deposit collagen is going to be down, right? So, we were curious whether, this was going on with smooth muscle cells in culture. So to do this, we did a matrix isolation technique. So basically, we take cells in culture and we separate the DOC soluble, deoxycholate soluble cellular fraction from the insoluble matrix fraction. We can then, separate these out and do Western blotting, or we can stain the matrix to see what all is there. And what you can see is that when we hit our cells with serum, this is not labeled properly, but when we hit cells with serum, fibronectin deposition goes up. When we delete Fa2, fibronectin deposition is cut in half, right? And you see the same basic thing when you look at fibronectin staining down here. When we hit with serum, it goes up. If Fa2 is not there, it won't deposit particularly well. This did not have anything to do with expression. We see slight trends that mimic it, but not to the extent that we would expect. And we don't see any changes in any of the proteases. We don't think it's the degradation. So we were curious if we're not seeing a big induction of, or a big change in expression or a big change in degradation, maybe it's the machinery that deposits fibronectin that's getting changed here, right? So if you look at how fibronectin is deposited by the cell, you get the formation of these structures called fibrillar adhesions. So normally you'll have integrins such as the fibronectin binding integrin alpha 5 beta 1 bound to talin here on the outside of the cell. As the cell binds fibronectin and pulls it into fibrils, you'll get the formation of this different type of adhesion called a fibrillar adhesion that's rich in alpha 5, but it's also rich in this protein called tensin, right? And these tensin alpha 5 rich adhesions actually pull fibronectin into the fibrils that people are used to seeing. So when you look at the isolated integrin adhesions, we see a reduction in alpha 5 and in tensin when Fa2 is not there. If we stain cells for tensin, you can see cells that express Fa2 have these nice long tensin adhesions, which are largely lost or considerably shorter when Fa2 is not present. we looked at this a couple other ways. There's some antibodies that will bind to the activated alpha-5 and beta-1 integrin. So here you can see active beta-1 in these long adhesions here. Same for alpha-5. When you delete Fa2, those adhesions are lost. And this kind of suggests that as Fa2 expression is coming up in these smooth muscle cells that are undergoing that transition, their ability to deposit fibronectin and potentially migrate out of the media into the plaque is reduced. So we didn't see any change in alpha-5 expression, alpha-5 activation. So we were curious, how Fa2 was mediating this. This is an active process that requires contractility. So we were curious maybe if this is affecting smooth muscle contractility to affect the process. So we put cells in gels of rat tail collagen, allowed them to contract it overnight. And what we saw was that there was a significant reduction in the contractility or the contraction of the gel when Fa2 was gone. We thought, well, this is suggestive, but there's a lot of reasons that this may happen. One, we showed Fa2 is involved in proliferation. Maybe there's just less smooth muscle cells there. So we looked a little bit more directly by just looking at myosin light chain phosphorylation within these cells. And what you can see is that, if we do a Western blot or if we stain for phosphomyosin light chain, Fa2, depletion significantly reduces myosin light chain phosphorylation in these cells. So this typically occurs by a couple of different mechanisms, one being activation of calcium calmodulin signaling, which activates myosin light chain kinase. But when we looked at calcium flux with the Fibrillar, we really don't see any changes. The Fa2 knockouts look just as capable at inducing calcium signaling. The other pathway is the activation of the RhoA pathway, which actually turns off myosin light chain phosphatase, as well as ROT can phosphorylate that directly. So we use a GST rhodokin pull down to look for the active RhoA, and we can see that that's increased with serum in the Fa2 expressing cells, but when Fa2 is gone, that induction is gone, suggesting that Fa2 expression promotes RhoA, but not necessarily get calcium. So this was a little bit confusing because RhoA is classically involved in ligand-dependent signaling, right? So normally what's known to happen is when Fa2 is ligate-gated, we'll get activation of RhoA, but we're stimulating this part of the pathway with FBS to get ligand-independent signaling that's also being associated with this. So we were curious why that would be. So we looked at the different phosphorylation sites that's induced by treating with serum. We saw 772 go up, we saw 897 go up. And when we stain the smooth muscle cells for phospho S897, it's localized pretty much throughout the cell, right? It's fairly uniformly localized. But 772 appears to localize 2 sites of cell matrix adhesion. And this co-localizes with active focal adhesion kinase here. When we look at isolated integrin adhesion fractions, we see Fa2 coming up, and we see that 772 site coming up. So we think that there's a ligand-independent, potentially, phosphorylation of 772 down here within the focal adhesion. To see if this was truly ligand-independent, we expressed a ligand or ligation deficient mutant R103E in Fa2 knockout smooth muscle cells, and you can see, yeah, it ligates it. It stimulates 772 in the focal adhesion just fine, right? So it doesn't require ligation, and if we inhibit Fa2 activity with ALW, it induces it even more. So it's not dependent upon Fa2. So we were curious whether this was mediating some of the fibrillar adhesion effects. So we took Fa2 knockout smooth muscle cells, and then we rescued back in either wild type Fa2 or Fa2 lacking that phosphorylation site. And what you can see, let me actually get through this and blow it up because it becomes a little bit easier to see. Let's see. There we go. So you can see in the insets the cells that are expressing Fa2. So, in the wild type Fa2 rescued cell, you can see that there is an increase in myosin light chain phosphorylation in that top middle panel, whereas the cells around it still show very low levels of myosin light chain. 772 rescue doesn't really induce the same effect. If you look at tensin fibrillar adhesions, the cells that are expressing Fa2 are shown in white there in the inset in the middle panel. And those cells that are expressing Fa2 show a nice induction of fibrillar adhesion formation, whereas 772 does not. And the same with fibronectin deposition, and 772 doesn't really do anything there. So we think 772 phosphorylation within that focal adhesion is critical to mediate this effect. And it's known that integrin adhesions can turn down Rho signaling through this protein P190 Rho gap. So we were curious if Fa2 and the focal adhesions was kind of turning that pathway off. So we looked at phosphorylation, and in the, when Fa2 is gone, the activity of P190 Rho gap goes up, either when we delete it or we inhibit its kinase activity. And this would be expected to reduce Rho signaling in the knockout. And just to validate that this is what's mediating the effect on fibrillar adhesions, we knocked the P190 Rho gap down. And you can see in the Fa2 knockout smooth muscle cells up here, very small adhesion sites. And then when 190 is deleted, these sites start to elongate into more fibrillar adhesion type structures. And when we knock down P190 Rho gap, we get a nice induction of fibronectin deposition. So the summary of part 3, when Fa2 is not present, like say in the quiescent smooth muscle cells, the integrin signaling likely through fat turns on P190 to limit Rho activity, limit contractility, and limit fibronectin deposition. But when Fa2 is expressed, we get phosphorylation of that 772 site, which turns this off, allowing Rho-mediated contractility to pull these adhesions into fibrillar adhesions and to promote fibronectin matrix deposition. So, in the context of treating athero, I think this, has some potential implication. So the inflammation arm of the pathway is ligand dependent, right? However, the other aspects with smooth muscle proliferation and matrix deposition, those are ligand independent responses. So if we're talking about taking a vulnerable plaque and pushing it more to a stable phenotype, we would want to inhibit inflammation and we would want to promote smooth muscle incorporation and matrix deposition. And what this data suggests is that if we can inhibit Fa2 ligation or Fa2 kinase activity, we should be able to reduce the inflammatory arm of this response while leaving this part of the pathway intact. All right, so since this is a cardiometabolism seminar, I figured I would tease with one metabolism story here at the very end. So one thing that we did notice in our Fa2 knockout mice was, even though plaques are smaller in the knockout, cholesterol levels typically were higher, right? And that was both looking at, cholesterol, triglycerides, and it was both on the Western diet and when we just put the mice on chow, right? So there was something going on that was raising cholesterol levels. So we're curious whether there's a role for Fa2 in the liver. So Fa2 is upregulated in the liver in response to inflammatory stimuli, LPS, and ischemia reperfusion injury. A GWAS study had identified Fa2 as a gene associated with circulating liver enzymes, and the SNP is actually associated with Fa2 expression within the liver. So we were curious what was going on there. So this work was picked up by Brenna Pearson, a graduate student in the lab. And she looked at Fa2 wild type, Fa2 knockout mice, fed a high-fat diet. And we see that the knockout mice actually show less weight gain. They show less liver weight, and they show a reduction in the liver weight body weight ratio, which is markers for non-alcoholic fatty liver disease. So when we looked for, liver histology, this is just a high-fat diet treatment, so we're not expecting, full-on NASH, but we do see some hepatic steatosis and inflammation within the wild-type mice. So we had these scored by a pathologist, and the wild-type mice all showed very severe steatosis scores, whereas the Fa2 knockout, half were severely steatotic. About a third, I guess, were mildly, and nearly a sixth had very little to no steatosis. If we look at inflammation and hepatocyte ballooning, both are reduced when Fa2 is down, and the overall histology score shows the Fa2 deletion reduces indices of non-alcoholic fatty liver disease in these mice. Very recently, MedRx 2021, a paper came online showing a GWAS analysis of non-alcoholic fatty liver disease genetic loci, and at or near the top of their list was Fa2, right? And they associated this with metabolic genes. I would probably suggest that Fa2 belongs over here in the metabolism / inflammation genes. And the S&P that they identified also correlates with liver expression of Fa2. So this is more early non-alcoholic fatty liver disease data. We also established a collaboration with some physicians down at Ochsner to look at patients with NASH, right? So non-alcoholic steatohepatitis, which have a lot more fibrosis in their liver, right? So these highly fibrotic livers also showed some changes, but it was not quite what we observed in fatty liver disease. We actually see a decrease in expression of FRNA1 in these patients, a decrease in indices of ligand-dependent signaling, that 588, right? But we see a trend toward an increase in 772 phosphorylation and in 897 phosphorylation, right? And this is consistent with the proliferation effect of Fa2, as well as the matrix deposition effect of Fa2, consistent with a potential role for Fa2 in fibrosis in late-stage NASH. So with that, I will acknowledge the OR lab members that worked on this: Brenna Pearson, Matt Scott, Stephen Funk, and Alex Finney especially, as well as our collaborators here at LSU, as well as elsewhere. My funding, and I'm happy to take any questions.
Hey, great, thank you. Did we lose Lale?
I think so.
Does she know that we lost her? I don't know.
I think she's still talking. No. So who has a question?
Like Muriel.
Yes, hi, can you hear me? Great, great talk. I absolutely love the microscopy images. They are beautiful. So my question is, what is driving the expression of F2A, the increased expression in the smooth muscle cells in late atherosclerosis compared to like earlier stages? Do you think that this is in response to lipid loading of the smooth muscle cells, or like macrophage derived factors that are secreted and increased in the advanced plaques?
So I think there's probably multiple answers to that. We were really hoping it was the lipid loading thing, right? Because that would have been a very, very cool story. We really haven't seen much data for that. You know, everything that we've done has come back negative. We have a hard time causing the smooth muscle cells to take on any macrophage-like gene expression properties either, right? We haven't had much luck with that. I would say based on you know, what we know in endothelial cells, it's very, very likely that some of the pro-inflammatory cytokines that may be released by the macrophages, T cells that are present within the plaque may be feeding into that. We're currently looking at that now. In the presence of serum, right, in our cell culture model, which is the easiest one to answer that question with, it's not anything we would have expected. We don't see any growth factors doing it. I was expecting PDGF to do the same thing. Doesn't. Surprisingly, the only thing that has mimicked that effect is the fibronectin that is present within that serum. So if we deplete the serum of fibronectin, the ability of serum to induce Fa2 is gone, right? And if we inhibit the fibronectin binding integrins within the smooth muscle cells, the ability of serum to induce Fa2 is gone. So we think that there's, you know, I hate feed forward pathways in biology because, you know, those are problematic, right? But we think that the early expression of fibronectin and the smooth muscle cells seeing that may pick up Fa2 expression to promote its deposition, right? And that kind of feeds back into the nature tree model.
Okay, thanks.
Katie?
Yeah, that was really great. Oh, Lale's back. Okay.
Sorry, I was kicked out of the Zoom. Yeah, I have a question, but I can wait for the others.
Sure, you can go next, Lale.
Okay.
So I was curious about that liver phenotype, and it sort of reminds me of our talk, was it last week or the week before, by Ali Cohan about, you know, what's good for the heart is bad for the liver, you know, I guess in vessels versus liver. So you see, as you describe, you know, pro-fibrosis in this setting of, you know, potentially the plaque, which is good, versus in the liver, it's bad. So then extending to that, have you looked at the heart and seen if there's any signal around ephrine, you know, associating with any kind of fibrotic phenotypes in the heart?
We have not, but that doesn't mean it hasn't been done. There was some work on the cardiac stem cells that was a little bit iffy. I forget the groups, and they suggested a role for Fa2 in the recruitment of cardiac stem cells into the heart, into the MI lesion, I believe. Another group has recently shown that if you I think I'm remembering this correctly, if you delete Fa2 out of the heart, cardiac fibrosis is down, right? Which is consistent with what we're seeing in the plaque. So I think, you know, with fibrosis, we're going to have to get rid of its expression. Just blocking its kinase activity or blocking its ligation is not going to do a thing. But we're going to have to do something to reduce its expression if we're going to get fibrosis to go away. Inflammation, we may be able to block it. Yeah, so I think it does have a role in the cardiac fibrosis too.
Thank you.
Can I go? Okay, great. So Wayne, I'm going to ask a question which is in my safety zone. So you have shown that this Fa2 knockout mice also have less weight gain, right? And that, you know, in metabolism, that plays a big role in, you know, that most of the time correlates with liver weight. So do you think this may contribute to some of the phenotype? I'm sure you're planning on doing liver specific knockouts, right?
Yeah, so we developed a conditional. We're going through the cell type specific knockouts now, as well as AAV mediated deletion and overexpression in the liver. we think it very well might, but we're not far enough down that road to have a definitive answer.
Okay, great. And a quick question. Do you see any glucose phenotype in these mice as well?
Yeah, we were curious if we would, because there was an early paper suggesting a role for, I don't remember if it was general FA or specifically FA2 in pancreatic beta cell insulin secretion. So our early studies, when we were feeding high-fat diet and the FA2 was knocked out, we were doing glucose measurements, just to see. We didn't see any such thing. So I don't think in the context of an ApoE double knockout that we're going to see glucose changes. If we go back and do that with a wild type, for ApoE, whether that would change things, I don't know. It's possible.
Okay, great.
Esther?
Yeah, that was a great talk, Wayne. I really enjoyed it. I was curious, you did all those bone marrow transplants and then you conclude the stromal fraction is probably more relevant. Have you also considered that the EPH2 is present on resident macrophages that are not hit by your irradiation? Do you know if that's present? It's just out of curiosity. I don't know the answer. So
very well could be. We haven't gone so far as to try to isolate those cells out to see if they're expressed. Looking at our that it seems like that would be hard to pick out by staining alone, because whenever I stain vessels, I hardly ever see any macrophage staining. But we haven't gone so far as to try FAXnet analysis to pull it out.
okay, cool.
Ada, did you have one?
Yes, thank you. I might have missed this, but I remember one of your first slides, you showed that the Eprin2 is upregulated when the smooth muscle cell actin is downregulated, right? When you start losing the expression of smooth muscle cell actin.
Yep.
And I was wondering if you have any data or are planning to do any, fate mapping experiments to kind of understand if those cells are the ones that are, converting to be macrophages or the ones that are proliferating in plaques.
We got Gary's lineage tracing mice way back when. That's probably one of the things that have slowed down our macrophage or our smooth muscles, specific knockout more than anything else. Just back crossing everything into the smooth muscle tree, the lineage tracing, and the ApoE. Those mice are starting to come down now. So maybe the next time we have an actual meeting, I'll be able to answer that.
Looking forward to that.
Okay, if there
Lani, can I have last question?
go for it.
Wait, nice talk. I'm just wondering, do you know if your EPH2 expressed in hepatic stellate cells, which is the major cell type drive liver fibrosis in the liver?
Yeah.
Stellate cells.
We think it's likely to be expressed in the hepatic stellate cells, but we have not definitively shown that, right? I would be surprised if it's not, but we haven't definitively proven it.
Okay, cool. Thank you.
Hi, I also have a question.
Okay.
Yeah, so related to your body weight gain differences, I was wondering if you have had a chance to look at their adipose tissue.
Yeah, I haven't, and I was waiting for that question because that's an obvious one, right? That we may have change in the edge, and we haven't looked. That's not in our wheelhouse, and that's part of the reason why we haven't looked. But it's definitely something that's worth doing.
Okay, thank you.
All right, great. Thank you, Wayne. Thanks for an amazing talk, and thanks for the great questions. I think there's this trainee session together with Wayne. Thanks, everyone, and we'll see you next week. Bye.
Bye, everyone. Bye, Wayne.
Hi, see you, Katie.
Ada, I'll make you host.
Thank you.
Okay. Thank you, Wayne.
Yep. Yeah.
I already made my coffee for the happy hour. I don't know if anybody wants to go grab some beer or coffee or anything. But I really also appreciated you sharing, the statistics of how, you got that funded finally.
Yeah.
You only see the successes, right?
Exactly.
So as a trainee, like I really, really appreciate understanding of the whole process and that it's not easy for everybody.
No, and it's one of you never really know. Some projects I think I'm going to get major pushback on, and I get a good score straight out of the gate, right? And some projects that I really like and I think are really interesting, and I cannot get reviewers to agree. So yeah, it's a crapshoot. Just submit as much as you possibly can.
Yeah, but then you also have, those situations where you get a very good score, maybe not funded, but very good. And then the next time you're like so sure that you're gonna get it, and you get
worse.
Bunked.
Yep. Yeah, I've had that as well.
Before I leave, I want to say I joined that club of getting worse on the second submission, and
yeah,
I didn't see it coming. It was
it's
like.
Yeah, after a 14 percentile to go to not discussed, and then to go to not discussed again whenever I sent it back. Yeah, that was a hit to the ego.
So any tips about, as trainees, like what should we do? I mean, sometimes maybe it's just not a good project and you don't know it, like you love it so much.
Problem, right? You don't know if. I think, with me, it was just a matter of, I really believed in this project, right? I thought that there was definitely something there if I could just get if I could just put what's in my head on the page, right? Then, things would work out. But yeah, knowing when to stop throwing money at a bad project is an important skill set, especially early on when the amount of money that you have to get your research program up and going and developed is limited, right? You've got to make some really tough choices on, here's my first priority, here's my second priority, and I'm just not going to work on this thing. And then you see that come up in Nature in 3 or 4 years, and you're right.
Yeah. So I want to encourage our trainees to turn their camera on and ask questions and introduce themselves. By the way, I'm a postdoc fellow with Ed Fisher, looking to transition whenever somebody hires me.
We're aware, and we're about to start hiring again.
Oh, that's good.
Shreveport's quite the culture shock compared to New York. I think art is one of the exceptions rather than the rule there.
Yeah, I came to New York from Israel because I said I want a mellow place, so somewhere to relax. So I went to New York. Maybe now I can transition to a truly mellow place.
Yeah. So any questions from the group? I know, whenever I was a trainee trying to figure out how to make that transition, I had a ton of questions, and I sought advice for wherever.
I have a comment. I think I have a friend who's starting at LSU, Oren Ram.
Yeah. So he, Oren Ram, was hired into our department, starts in May. Looking forward to having him here. You can see with our non-alcoholic fatty liver disease project, we need more expertise.
Yeah, I was just thinking that.
So yeah, I'm hoping he comes in and helps us out.
I do have a more practical question. So when you're first starting in your lab, how do you balance like doing experiments or writing?
Yeah,
I've heard it both ways, where people are in the lab a lot in the first 2 years or so, and then other people
I was in the lab,
don't want to touch a pipette.
No. That's you're your best employee for the first several years, right? It's hard to get people that know what they're doing coming in, because you guys are going to be new, right? And you're competing for postdocs with Ed Fisher and Eugene Chen and whoever, Ira, all of them, right? So it's hard to get people that are know exactly what they're doing coming in. So be prepared to train, right? And the only way to really train people and to get them to do things like you want to do them is to be in the lab. So for, I guess, up until I think right after Mabruka started, it's like 2016, I had a bench and a desk in the lab, and that's where my computer was, and that's where I sat all day long up until 4 or 5 years ago. Since taking over the cardiovascular center, the admin work has just become too much, and I just can't do experiments anymore. But from the time I started in 2007 until 2016, I had a bench, and I did experiments, and I helped to push projects forward. So yeah,
interesting. Yeah, thanks.
Stay in the lab as long as you can, right? Eventually, everyone has to stop, and when you do, your skill set, tanks. So take advantage of the skills that you have as long as you possibly can.
What's the proportion of time that you spend in the lab as a junior investigator?
I spent probably 50 to 60% of my time on experiments and 40% of my time on admin. I mean, but the amount of admin is going to be a lot more than you guys realize, right? Not even stuff that like say departmental duties or teaching or any of that. Just getting like biosafety protocols up and going, right? The IACUC protocols, any modifications to those things, just random stuff here and there that take up so much of your time when you're the PI that you never see as a postdoc, right? You just see the PI pop his head into the lab for a few minutes here and there, and not realize how much of their time is just bureaucratic nonsense to keep the the lights on.
Yeah, I mean, I'm kind of dreading that point.
It's fun because it's finally yours, right? I think there is a time in most postdocs' existence where they get real tired of doing experiments that someone else gets credit for, right? It's like, you go to a meeting and you're like, oh, Ed, it's beautiful work. And you're like, damn it, that was me. That was my late nights. So yeah, eventually you get really, really frustrated with that as a postdoc. So, having the keys to the car, I think that's worth all of the admin burden that comes out of it.
Can you maybe address like what specifically you like about the institution that you're in?
Oh yeah. So there are a lot of things. So when you're at your stage, right? A lot of people don't understand the difference between soft and hard money, right? So a lot of private institutions will hire in faculty on soft money, right? Meaning that the institution really only guarantees a very small percentage of your salary. The rest of it you have to cover off grants. Whenever I was at UVA working with Martin, I thought that was just a model, right? I thought that was the only way that it happened. It's not true. There are places that you're on hard money, at least a large portion of your salaries on hard money. Here, I'm 100% hard money, right? But it's made up for. My salary's a little bit less than what it would be if I was at one of these institutions that makes you put everything on soft money. But if I perform, right? So we have what's called a research incentive compensation plan. So if I cover 60% of my salary off grants, right, which is not, it's not huge compared to what people are being asked to do at other institutions. If I cover 60%, I get to give myself a 30% bonus, right? So I give myself a raise of 30%. So if things don't go well, you have a hard time getting funding, you're not screwed, right? You don't run into the issues of, if I don't hit on a grant within the first couple of years, I'm going to be out of a job, right? You have a little bit more runway. But if you do well, you can, have your salary be what it would have been at one of these other places. And I'll tell you, the cost of living in Shreveport is way different. So the salary, it goes quite a long way here in Shreveport. The issues that most people would think of when why they wouldn't come to Shreveport would be, I won't have access to the facilities, I won't have access to the collaborators, right? So Shreveport is a small place. However, it does a good job of understanding that it can't be good at everything, right? So we put all of our resources into two areas: cardiovascular research and cancer, right? We even have dedicated endowments that support cardiovascular research and cancer research on our campus. So the Cardiovascular Center has a dedicated $2 million coming to it every year that we can use to fund intramural grants, pre-doc fellowships, post-doc fellowships, core facilities, et cetera, et cetera, et cetera. The difference between the core facilities here and the core facilities that I see elsewhere is that the core facilities here, the technician's salary is covered off the endowment, right? Which means that the amount of money we have to charge to keep that core facility open is just above cost, because we don't have to charge enough to cover the technician's salary. So the core facility is there to enhance research on campus so we can keep everything at just above cost. So it is way, way, way cheaper. Like say we just started up a surgical cord not too long ago for a mouse MI surgery, right? I think I saw at a couple other cores, those are in the hundreds of dollars, right? Here it is either 40 or 50 bucks, right? So like partial carotid ligation surgery, that's I think $30. So I mean, we have an AAV core that produces our like AAV PCSK9 that everyone is starting to use more and more of. The difference is that instead of $3,000 for a batch, we charge our internal people $600 for a batch, and that is enough for I think 200 mice. So it's the cost of doing science here is very, very low. We've got a good number of strong cardiovascular people to work with. Core facilities are actually excellent. We actually have the ability to access those core facilities because they're not booked up for the next two months. And there's support.
So you can maximize the experiments that you do, the amount of money that you get, right?
Like our animal costs are like 50 cents a cage, right? So I would be paying another 20 to 40 grand in mouse costs a year at some of these other institutions, and I can take that money and hire in another student, right? Do
probably a month, not a year.
Yeah.
Yeah, that's probably what we pay a month in our animals.
Yeah. So yeah, it's it's a good research infrastructure here.
May I ask a question?
Yeah, go ahead.
Hi. So thank you for the organizing committee first. So I'm asking, as Mark Nawi, I give the first talk, and thank you for being inclusive for including our cancer research work. I thought I don't have a chance, but when I got there, I was surprised. So thank you for that. I just want to ask about the fabulous tips that you're giving. How can you find those information? Because what you meant is that big place is not always good because the amount of hidden costs and also trainee in transition. What is the best steps that they can take, especially at the right time?
Right. So this is a big part of it, right? Networking, getting out and meeting people, so that when it does come time and you're looking around, you can, send an email to someone at that institution and say, hey, I'm looking around. Can you let me know what's available at your place? Especially I'm, interested in this type of microscopy or this type of small animal imaging or something, and what's available, how much does it cost, tell me a bit about the department that I'm thinking about applying to. So networking is huge in this job, absolutely huge.
Yeah, probably any job.
Yeah, I think even more so this one, just because when it comes, at the end of the day, our grants are reviewed by our peers, right? And the grant success is the best determinant of how our career goes. So I don't think being a good networker is going to get your grant a better score from reviewers 1, 2, or 3, right? I think reviewer 1, 2, and 3 are going to review that grant to the best of their ability and to their content. What networking does, it affects the other 22 people in that room, right? So, you've now got a grant that's got one reviewer saying 2, one reviewer saying 4. If the other 22 people in that room know you, like you, think you're a decent person, and want you to do well, their inherent bias is going to go, I hope the one that's saying it's a 2 is right? Versus if they don't know who you are, right, they may be more tended to go in the middle or go to a 4. And the difference between, a 20 and a 40 is funded or not, right? So, I think networking is important. It's not as direct as I think some people think, that if you meet the right people, your reviewers are going to go easy on your grant. They're probably not. They're people try to review grants as honestly as possible, but you're trying to get the rest of the room to like you enough to want you to do well to get you a good enough score.
Any other questions from our trainees? And do you have any other tips? I mean, we have also some grad students here, and junior postdocs, so maybe some tips for them.
Yeah, I mean, if once you go to look for a job, right, whether it's going from grad student to postdoc or postdoc to faculty, understand what's driving that, your papers, right, and your grants. Whenever I'm going through a list of faculty candidates, that's the first, I don't look at any of the other superlative stuff, right? I go and see, what are their papers? What are their grants? Because past success is the best predictor of future success. So if someone was highly productive during grad school and during postdoc, got a pretty good indication they're probably going to be highly productive as a faculty. If they publish sporadically as a postdoc or grad student, then I have to worry, all right, are they really going to have the amount of productivity as a faculty member that are going to allow them to hit on a grant and succeed? So for grad students, make sure you publish, right? Don't rush yourself out. Get a good number of publications under your belt before you rush out of grad school into postdocs. Postdocs, publish. you can collaborate, right? You know, a high number of publications is good as long as you have a decent number of first authors, right? So you want to make sure you're publishing your own first authors, but as long as that's taken care of, you can also get a couple more out of collaborations. You're going to be better off. And then understand that, you know, the people that are doing the hiring, those are people, right? There are people that are being given the opportunity to make a assessment call on whether this person is going to be good, and no one wants to screw up, right? So they're going to give the position to the person that is the most likely to come in and do well as a faculty. And, you know, the best indication of that is some sort of funding coming in. You don't have to have an R01. You don't have to have a K99 R00. If you do, that's great, right? But you don't have to. But having some sort of funding is helpful, whether it's a, you know, a transition grant, you know, AHA Career Development Award, you know, ADA Junior Faculty Award, something, right? You know, whenever I was looking for jobs, I published well. I published real well. And I started sending out applications and nothing, right? Nothing for like 6 months to a year, nothing. And I contacted some of these institutions and I was like, you know, let me know, you know, one, where I am, you know, what was wrong with my application? Was there anything that I could have done better to make me more competitive? And, you know, the answer I got back was, you know, we really like you. You made our short list, like our list of like 10, but you didn't make our short list of like, you know, the top 3 that you're going to bring in and interview for the job. And then I got my AHA Career Development Grant. And after that, every single application that I sent out, I got an interview for. So, you know, just having that transition grant, you know, made a huge difference for how my applications were viewed by those committees.
Yeah, I think that one of my major problems is the work I did with my PhD, because my boss retired 2.5 years into my PhD.
Yep.
And I had to leave within a year, basically.
Well, but you know, recent productivity can offset that, right? If you're publishing a lot in a postdoc, then you know people will give you a pass. And
I hope so. I mean, for now, like the last hiring cycle, I got like 0 interest, but we'll see in the next one.
Yeah. Good luck. We're expecting to post a position in my department later this month.
Ah. Thank you. Good to know.
Not saying you want to come to Shreveport, but any other questions from the group?
Yeah, I had a question about going back to the topics of, you know, things that you should know when you're starting your lab. Yeah. It's about choosing the right project. Obviously, in the beginning, you talked about, you know, not everything is fundable and there are ideas that you love, but you know, you might have problem convincing other people. I was wondering, what would be your advice for a new faculty on, do you want to do big things, exciting, you know, groundbreaking, expensive, or small things to just get some publication and then?
Everyone's got a different take on that, but my take, to use a baseball metaphor, is to hit singles, right? You know, just start being productive, get things out, get papers out. So as far as choosing projects goes, do things that you can do, right? You know, you've got a limited number, you know, limited amount of money from your startup that you can invest in getting the resources you need for a grant, right? You're gonna need a ton of like mechanistic insight. You're going to need new animal models, most likely, right? You know, if you do some, you know, RNA-seq, single cell seq stuff, that's very helpful. The, you know, the committees love that kind of stuff, but it's got to be paired with mechanism, right? Otherwise, they're going to come back and saying it's superficial and descriptive. So, you know, it's getting a project up off the ground enough where it's going to be potentially fundable is not trivial. So, you know, you've got to look at, you know, what is my activation energy for this project? Am I going to have to create a mouse from scratch? Or is there a mouse that is currently available either through a collaborator or online that just hasn't been used in my, you know, disease model, right? You know, are there, you know, is there experiments that you can do that will, you know, fit into a couple different grant ideas? So just, you know, coming up with what's the best use of that money that's going to get you the easiest, you know, next grant. And with, you know, with the R01, put a lot of your focus into your main project for that R01, and then, you know, don't be afraid to send some of your smaller projects out to American Heart or, you know, some other, you know, groups that may fund smaller projects. Now, I'd say it took me 10 years to get the Fa2 grant funded. I mean 10 years to get it R01 funded. I had got a AHA funding in 2015. I think 2017, a grad student got a postdoctoral or a predoctoral fellowship on it. So, you know, I've had little bits of things here and there to help push that project forward, keep it going. But, you know, it took a while to develop before it was, you know, competitive for R01. So, yeah, I mean, just you really need to sit down and go, all right, here are my best ideas. Of those, here are the ones that are most doable, right? And that, you know, that would cost me the least amount of money to get up off the ground, and then just, you know, base it on that. Ultimately, you know, it's your call. You know, it's your shot to get, you know, your career up off the ground. So you got to go with whatever you believe is going to get you there. So, you know, it's the last thing, I guess. Don't work on a project just because it's easy. If you don't find it cool, then don't bother.
Thank you. That's great advice.
All right. Any other questions? Okay. So thank you so much, Dr. Orr. This was really, really helpful.
Yeah. And if you guys reach out for any more questions or, you know, concerns, don't hesitate. I mean, I don't know if you can tell, I don't shy away from giving advice. So please reach out.
Thank you so much.
Thanks.
And thank you for the seminar also. It was fascinating.
Thanks.
All right. Have a nice weekend, everybody.
Bye.
Thank you. Have a nice weekend as well.
Bye, and keep joining
Bye.
us, guys. We will see you every single networking session.